Hello, this is George McNamara. I'm going to give a not so brief history of spatial biology. My original presentation at BioTrac at Montgomery County College in Maryland back in September 2024. I'm now recording this in January 2025. I'll include the current my BE Press website, web links for the presentation, 100 plus slides, and the video, which I'm going to repost after I get this one done, since the previous one cut off, and I apologize for that. I want to start out with this, what I consider the classic stock market drop on the part of Bristol Myers Squibb for screwing up a percent positive cut point in one of their Opdivo anti-PD-1 clinical trials. Their stock dropped by $22 billion in one day, about the 20% market value. Merck and Company, U.S. Merck and Company stock went up a little dip on that day, even though Merck had no news. Don't let the front office, the CEO has been, you can go find the articles in Forbes about the CEO basically guessing wrong on the cut point, probably overriding their pathologists and oncologists, whereas Keytruda had a much more conservative cut point. And ever since then, Keytruda sales, these are almost identical anti-PD-1 molecules. They almost certainly work by the same mechanism of action. They probably cost about the same to manufacture, billions of dollars. Their CSO at the time bet the company basically on the anti-PD-1 and then ideally more immune checkpoint blockade monoclonal antibodies and tweaks and other therapies will eventually, it is curing cancers, but it's not curing everybody. More checkpoints are going to infused in the antibodies infused in the same patient, going to cost more money. Going to cost more money to do the trials. Again, not everybody is going to make it through. Not everybody is going to get cured. And then, interestingly enough, either the same antibodies or sort of the complementary suppress the co-stimulation or find antigen-specific T cells or the right antibodies to kill off the bad B cells making antibodies may actually be a bigger market for autoimmunity than it is for oncology, for immuno-oncology. And so the information gotten out of the PD-1 and CTLA-4 and other trials may help cure or at least suppress the autoimmunity, and then ultimately the goal would be to kill off the antigen-specific B cells and T cells without suppressing everybody's immune systems against pathogens like COVID-19, influenza, various bacteria, various other things, toxins. There's cool things happening in terms of snakebite venom cures with antibodies or mini-bodies or AI-designed proteins, and a lot of good things happening. Spatial biology, and I'll give a sort of quote, more or less definition in a couple of slides, but informally, it's essentially all biology other than ATCG and sometimes U of molecular biology and biochemistry. And so it's kind of ironic that the word spatial needs to be in front of biology in 2024, and companies are raising tens and hundreds of millions of dollars to be in spatial biology niches. There's been some fallout of things like the owners, the same owners, for both Ultiview and Vizgen decided that they would be better together under one name. And so back several months ago, around the time of the BioTrac meeting, the two companies were merged together, and so like the same owners slam these two things together. Okay, whatever. They're the owners; they can do what they want. Presumably, one executive team moved on to other. The people on one executive team mostly moved on to other stuff, and maybe with some advisory time for a couple months or a year of their former company. Expect more consolidation, more companies disappearing, more AI marketing stuff of new things happening. Okay, so the big thing is oncology. We're seeing a lot of cool things, and mostly open data access is good. And then in the future, if a lot of my biology colleagues at Johns Hopkins University medical school seem completely unaware of GLP-1, and I'm like, huh? How can you not notice the news in medicine and biology? And so I'll suggest that spatial biology is both for pathogenic things of cancer, oncology, autoimmunity, of COVID-19, other infectious diseases, but also for the normal tissue. We got to know what's normal, and then there's a lot of variation amongst humans and a lot of details in the genome that are not necessarily completely encoded in the genome because of epigenetics and history and altered states and so on. Okay, I've already spent 6 minutes on one slide. That's bad. In terms of getting this thing done in a reasonable amount of time and a reasonable file size to upload onto the internet here. So, as I said, it was based on a short talk, 15-minute, that I overran. It's a whole lot of slides. My approach to this particular video presentation is this is not a little TikTok short thing, and I have deliberately posted the slide deck on my BE Press website, and I'll probably find some other places where I can post it. It is 100 megabytes or whatever, so not completely trivial. And Elsevier, unfortunately, going to schedule to shut down the BE Press website in a couple months, and so I've actually posted the original version of this video on a YouTube channel that I created. But then I realized that I cut off part of the video, and I'm re-recording that, and I'll see if I can swap out the web link for the new version. Okay, the BioTrac stuff. This was a terrific conference. It's limited in space to about 80 people, and there's 15 or so presenters, including me and Reagan Baird and other people in this Origins of Spatial Biology session. It's a good chance to network at a much smaller scale than SITSI or cell bio or neuroscience or experimental biology, blah, blah, blah. This is not a Steve Jobs style presentation. You can go back and look at the Edward R. Tufte cognitive styles thing to see how also that I'm not doing that kind of presentation either. Yeah. So it's been, at the time I presented, this was a decade after Keytruda first got approval for melanoma, for a type of cancer, and now there's a ton of stuff on that. I apologize, I kind of repeat the same topic here. That was the session. Since it was my task was to try to organize some kind of history, I said, well, when did spatial biology happen? One of my colleagues from when I was at MD Anderson Cancer Center nicknamed me PubGeo. It's both a compliment and to chastise me about spending more time with the literature than at the lab bench. And we can see how that did or did not turn out in terms of my bank account. I identified in 2020 a paper by Chakra, the PI on this project, the last author on this particular paper. I'd actually had spotted this and kind of featured it because I've known Lance Taylor for a long time. He's been doing multiplex immunofluorescence microscopy for a really long time. His colleague Albert Go has been with them for a long time. Jinty is a key person from GE Global R&D, and then the rest of the authors are probably mostly in Pittsburgh, along with Chakra and Lance. The stuff before this paper were things like space biology and French and radiology stuff. So I identify this is the first paper to use spatial biology in its modern context. Keith Wharton down in the bottom right there is a former BioTrac speaker. His quote is right there. I kind of love the idea that spatial biology is everything except a molecular biology. And then some more featuring of Chakra, and then some stuff from his colleague at Predex Bio. Back before Spatial Biology Conference at BioTrac was named Spatial Biology, Sean Downing, who was at UltraView and has since moved on to other companies you can check out, most people I mentioned in the slide deck and LinkedIn, had a beautiful slide that I've zoomed in to one T cell on the left and one tumor cell on the right and the immune synapse, and that's where the action is. And so when I look at spatial biology at low resolution, where a cell is 1 pixel, or in some of the methodologies with giant 50 micron beads, one bead is accumulating RNA sucked off of 5 to 10, maybe even 15 cells. And so I think that to really understand most the immuno-oncology, maybe the autoimmunity, and maybe normal stuff, you're going to see what the cell-cell contact is doing and what the signaling molecules are up to. Now, in principle, if the sample had not been biopsied out of a patient and then immediately fixed and processed, that hopefully that T cell would have taken its kind of shotgun blast of granzyme B and perforin and other molecules to blast off and kill the tumor cell on the right there through the immune synapse, after recognizing this the target cell as a cell that needs to be killed. And then I have examples on the right of various signaling molecules. I have at the top right the classic discovery of the immune synapse by deconvolution microscopy. Beautiful paper in Nature in 1998. I'm gonna try to not read my slide decks, by the way. And then more of the same of there's a lot of stuff going on, and now with things like 50-plex or 100-plex, 101-plex, a couple of 100-plex, immunofluorescence microscopy, antibody detecting either the protein or the post-translational modification, or sometimes the interaction between two proteins, that you can measure things quantitatively. It would help if people actually knew how to use the microscopes correctly, and the vendors set things up to be quantitative. We'll see later, I'm a big fan of tyramide signal amplification, but amplification does not imply linear amplification. It implies more signal, and if you do it right, little or no more noise. And so how do you quantify things like tyramide signal amplification, or hybridization chain reaction, or RNA scope plus tyramide? Not a big deal, because you're looking at, in the RNA case, single RNA molecules. You can count the RNA molecules. You're not completely sure that you've counted every single one of the particular message you're trying, or long non-coding RNA you're trying to detect, but at least that is dots, single molecule, and that's really where it would be nice if protein antibody spatial biology could be, rather than sort of negative plus of classic HER2/neu IHC, which is semi-quantitative, which is really not quantitative. So anyway, this is all signaling on the T cell side, and then featured in the middle of the slide are several of the immune checkpoint molecules plus TCR zeta or CD3 zeta subunit. Biology and especially immunology have multiple, any given protein may have multiple names. And you just got to deal with the fact that PD1 has a CD marker name that's sometimes but rarely mentioned, and even granzyme A and B have alternative names. This slide most summarizes the original 100-plex immunofluorescence microscopy by Walter Schubert and colleagues back in 2006. But that might have been just one field of view, and they claim that they did the controls and made sure all the antibodies work, and were targeting the right thing. I'm like, sure, but if you don't show the data in the main paper of the supplement, I don't believe you. And then the list of companies back a few months ago, before Visgen and Ultiview merged, is down at the bottom right. Bottom left. Again, the companies, and then a couple of the press release things. Bruker rescued NanoString from bankruptcy, and also had at some point acquired Acuity, blah, blah, blah. Oligophysique idea up at the top right. I'm like, yeah, maybe sequencing is doable and accurate and worthwhile. And then bottom right, the merger about a month after the BioTrac conference of Visgen and Ultiview. The original commercial method, so Schubert was pretty much an academic thing, although there was a company affiliated with them, was Gerdes and Fiona Ginty, who was a co-author on the 2020 Spatial Biology sort of starting point, multi-omics. And then I have a box at the top right, a paper from Tan-In-Se, a colleague of mine at University of Miami. I'm a middle author on the paper, and Sandro Santagata, who's been on probably 5%, which is a lot, of the spatial biology papers since 2014, that you can get a sense of spatial biology as a term is 2020. As a concept, stretches back, and even back further in terms of just immunofluorescence microscopy, which is mostly cell biology stuff. In many ways, my brief was supposed to give a brief history of spatial biology, and so looking online, I found 10x Genomics infographics here, and you can stop the video and check it out, or you can pull up the slide deck from BE Press or wherever else I posted online, or conceivably I could send it to people by OneDrive or Dropbox. You can multiplex all sorts of different ways. That's what's a whole bunch, not comprehensive, but close, of the many different fluorophore types that you can use. Down on the bottom left are some spectra. I'm a big fan of spectral imaging microscopy. I used to work for the Sky Company. I've added to this infographic a bunch of panels like the Thomas Reed et al. ASI 1996 Sky stuff at the bottom left of the infographic, and I added some single molecule RNA FISH, single molecule ISH at the top center, some Boltex and Tyramide. You can find easily enough the original 10x Genomics stuff if you want. And then I also included some pricing, and I recently found looked online to get the hybridization chain reaction of about $5 per slide, a little bit lower than the classic Stellaris FISH from LGC Biosearch that I used back in Houston, which in turn is much cheaper than the branch DNA based stuff from ACD Bio, the RNA scope. Some other tools and some rehashes of what I had. Part 2 of the 10x milestones. The bottom right is my favorite example of single molecule RNA FISH. I did not take that image. LGC Biosearch has it posted online. It's RNA Pol RNase polymerase 2 alpha, which is this is the dots from the mRNA of the major subunit of the RNA polymerase that makes mRNA, or pre-met mRNA that gets chopped up into mRNA. And so I love this as a sort of snake chasing its tail deal of the biology central dogma of DNA encodes for RNA, RNA encodes for proteins, proteins then can replicate DNA and can make RNA, and life is good, or life happens. And then at the center of this, I mentioned briefly expansion microscopy. I'm a big, of course, a fluorescence microscopist, and so expansion microscopy ultra cool in terms of a physical way to zoom out the specimen rather than using fancy, mostly expensive super resolution microscopes. A very recent paper of Trisco. You can never have enough acronyms. This was TRIS meets DISCO. Shows very nicely a way to one of the most recent late 2024 methods to optically clear a sample. In this case, a mouse brain. Here is actually pretty nice water. Here's a fuzzy, can't see the background grid in SSC, and here is a somewhat improved tris over the water, and then some nice examples across the slide of clearing and getting good uniform staining of this particular set of labels. If you don't do things perfectly, the label just accumulates at the outer surface. If you do it well, there's pretty much nothing special on the outer surface. The bottom edge may or may not be artifact. It may actually, it could be a whole lot of mallet 1. And you got to understand the sample and understand what's going on in order to get good data. Consistently, these papers often just show one picture of the successes, and so I look fairly carefully at the method sections and go like, how many times did they do this? How many mouse brains did they do it? Can multiple grad students and postdocs do it from the lab if there's a whole bunch of authors? Can the PI do it? Ooh, that's a good question in terms of looking at the method sections or asking if at a conference if the PI is the speaker. Have they been in the lab and done it? Okay, I'm a big fan of fluorescence microscopy. I've thought a lot about how to make microscopes be quantitative, and basically there's a lot of stuff that can go wrong. I'm not sure that the microscope companies have done a great job all around on making they have not really tried to make it quantitative. And then all of the vendors in the spatial biology world, they are not necessarily trying to make quantitative things. A lot of the data I see in spatial biology is basically plus minus. Was PD1 above some threshold on the surface, assuming that there's enough resolution to see the plasma membrane edge of the cell, of a cell, rather than just one pixel, one cell? Is it's above background? I'd actually like the units to be molecules per pixel or molecules per micron cubed. And that's not so easy, and then the microscope needs to be set up perfectly. It needs to be clean. The sample needs to be clean. The refractive index of everything needs to be perfect if you're using high resolution. A lot can go wrong. So don't trust the authors of papers. Ideally, the data be online and have great metadata and clear methods in the paper and the supplement, but don't count on that. A lot of things go into the various versions of spatial biology, and then there's a lot of dependent history of things like immunohistochemistry down at the bottom right. A critical invention was the laser, the classic James Bond scene at the top right. The invention of LEDs, and especially, and then later blue and ultraviolet LEDs. The blue LED won the Nobel Prize and is in the Blu-ray disc drives that you watch your videos on. Used to watch videos before streaming happened. And then there's a lot of very nice to terrifically priced detectors out there. Although all of the ones on this slide are actually for the visible and near infrared, and occasionally a little bit of UV. So let's say many of these detectors are subsets of the detailed variants of the detectors are good from, let's say, 380 nanometers to 880 nanometers, maybe 980 nanometers, maybe 1,000 nanometers for round numbers. And so what's called the NIR2, 700 nanometers to 1,000 nanometers, you can get okay data to pretty good data with the standard microscope detectors like CMOS, scientific CMOS, or now the quantitative CMOS. But you cannot do above 1,000, and then down below, say, 380 nanometers, most of these detectors crap out, and you can't see those. But there's no fluorophores down there because you can't see them, or almost no fluorophores because you can't see them, so it doesn't matter. But near infrared 2, which would be 1,000 nanometers to either 1,300 nanometers, right, 1,700 nanometers, 1,800 nanometers, 2,000 nanometers, whatever. You write the papers, you choose the definitions of these wavelength ranges. There's the classic silicon detectors like these CMOS. They crap out and are not absorbing photons in the 1,000 to 2,000 nanometers range. The visible range is more or less 400 to 700, maybe 750 nanometers. The UV, there's various UV, A, B, and C, and so on down below 400 nanometers. And you need the right light source, you need the right detectors, you need the right fluorophores. Okay. So there's a lot of technology. And of course, computers have gotten a lot faster in the decades that I've been doing computerized microscopy. I do want to feature one critical lab bench or autostainer or technology, which is antigen retrieval. Not every single antigen in a cell or tissue can be retrieved perfectly with a single protocol. That's a problem because you'd like to be able to detect any of these things, and so you got to be aware that if you antigen retrieve in certain ways, you may eliminate the antigenicity of some proteins or some carbohydrate, lipid, whatever stuff. And that's simply not going to be measurable or not going to be usefully quantitative. And then one of the kind of cool things about antigen retrieval, one of the better methods, or some of the better methods, involve microwave ovens, which are spin-off of the classic cavity magnetron, which in turn was a critical component to the success of radar in World War II. And so there's a lot of intriguing things. I kind of like this example of as an unprecedented achievement, which instantly reminded me of what polymerase chain reaction did for molecular biology and what PCR did for biology as a whole. In situ, I should mention, in situ PCR exists, but kind of fell out of, was a very tiny niche and fell out of favor. And so you could detect either DNA or RNA by in situ PCR, but it was not very popular, and now there's things like hybridization, chain reaction, RNA-scope plus tyramide signal amplification, and so on, that let you detect and just a whole bunch of oligos, single molecule RNA fish, or in some cases single molecule DNA fish, that you can detect things. Okay, so another critical component, this is supposed to be a history presentation, tissue microarrays. I was sensitized in mid-2024 by the fact that some of my lab colleagues here Johns Hopkins University had drawers full of the wrong types of cover slip thickness. And so I've been emphasizing the last 6+ months now that for most things, people should be using the number 1.5H cover glass, since they're probably using objective lenses that can be high resolution, and that essentially all of those are standardized for 1.5H, which is 170 microns thick, plus or minus 5 microns, which is a statistical thing. So occasionally that one of those boxes is going to have either one or a whole bunch of things that, let's say 180, 185. You can measure this through the microscope. You can image the top and the bottom surface of a cover slip after it's been set up using simplest to use reflection contrast mode, the mention in the bottom left of the text here. You can even see whether the cover slip is flat. The cover slip is usually facing down on a classic microscope slide. And if you use the wrong amount of mounting media, or you have a stupid label on one end, physical label on one end, things are going to get tilted, and it's going to be bad, or either a little bit bad or very bad. Marienfeld in Germany is the main manufacturer of these things. There's a whole bunch of OEM distributors with or without their own branding. Again, more stuff said talking about 1.5H. I'm a big fan of imaging dishes for cell biology. Scale things, conceivably people could maneuver tissue sections, or, and more sensibly, just stick things like half a mouse brain into one of these imaging dishes and image up. More on cover slips. I'm not going to read the slide, but basically somebody had a nice presentation in a month before my BioTrac talk with a whole bunch of text, and so that's kind of cool. I came across this a couple years ago of everything you want to know about objective lenses. The bottom right is how ultra-pure glass gets made, in particular for optical fibers. Beautiful story. You can read about the stuff in the Corning Glass work stuff, and you can go to the Corning Glass Museum in, not surprisingly, Corning, New York, and check it out yourself. Very cool. This is a big, thick book that I've been reading off and on, mostly off, on various light sources. Kind of cool. Thick, easy read. One light source, one chapter, sometimes same light source, multiple chapters. But the reference in the top left. And then I mentioned previously, expansion microscopy is ultra cool, and on the right, various optical clearing methods, of which Ali Erturk's is, I'd say at this point, the leader in the optical clearing method. And then Ed Boyden, featured at the top center there, is the PI of the lab that invented expansion microscopy. A bunch of other companies and academic labs have variant names, but the same concept of physically expand the sample. And Ed has a beautiful TED Talk explaining, showing off baby diaper powder material expanding when water is added. And so that's a variation of that. In fact, originally that material, or very similar material, is what was used for the first expansion microscopy paper. And now there's probably an expansion paper every month, at least one optical clearing paper every month. And optical clearing basically can make, let's say, an entire mouse transparent. So the largest tumor that I can think of is probably smaller than in an adult mouse. And so you can make things clear. Or many human organs can actually be cleared. Then how are you going to image that? More on expansion microscopy. I'm not going to read the slide, but it's faster than, say, confocal microscopy, and then for certain things has advantages over conventional wide field epifluorescence microscopy. One of my favorite papers was this Maillard et al. Paper that reported that for red and near infrared fluorophores, and in particular Alexafluor 610X, which is not a well-published dye, that first of all it's bright, and second of all, replacing water with D2O, deuterium oxide, enables the fluorescent signal to go up. And it turns out that water, in particular the OH group, is a really crappy suppressor of fluorescence, a quencher of fluorescence, and so switching to D2O improves the fluorescence of some fluorophores like size 7 here from being pretty crappy, about 20, let's call it brightness 20 here, to brightness of, say, 60. So I use the analogy of, would you like to get paid $20 an hour, or would you like to get paid $60 an hour, or even better, Alexa Fluor 610X get paid $132 an hour. And then assuming the detector is equally sensitive across all those wavelengths, that and the same power light source for each of them, that you'd be better off with Alexa Fluor 610X in D2O than Psi 3.5 or 740 or ICG, which is ICG is one of the few clinically approved fluorophores for angiogenic, for blood vessel labeling and retinal angiography. I'm not saying that Alexa Fluor 610X should replace it for those clinical applications. It's not feasible to infuse people full of D2O, but reading the literature can get you insights in terms of how to find the best fluorophores. And then this is all in solution. So then the next question is, how well does Alexa Fluor 610X work when it's stuck on an antibody? And that's I don't know, and I wish Thermo Fisher would put some antibodies on, would put Alexa Fluor 610X on some antibodies and say, the alternative to Alexa Fluor 594, spectrally they're almost the same, maybe we should try those out. And then at the bottom center here, one very nice example from Glenn Boytke, sorry if I mispronounced that, of a size 5 variant that if you put the right linker on, that you can get a whole lot more photons out. And let's say that without a linker, size 5 would be at 100,000 photons as an average. A single molecule would be under these conditions before it photobleaches or photodestructs, that this would be 600, over 600, about 700,000 with the right trick chemical stuff. Tris NTA, nickel NTA is the chelator. Nickel is triplet suppressor if you have it the right distance from the fluorophore. It also suppresses fluorescence if it's too close. And so a certain amount of work that I'm going to guess would probably double the price of the fluorophore. If it gets you from 100,000 to 700,000, that's a 7-fold improvement. But if it gets you from, let's say, 10,000 to 700,000, that's a huge improvement. And then if it also turns out that D2O improves things by another 10% to 30%, that all of those optimizations make for better fluorescence, and maybe we should be doing that. This again is a history thing, so I also found a timeline of a whole bunch of different fluorophore classes and stuck that in. Munan et al. 2024, Chem Communications. Luke Lavis has also done stuff. The classic Alexa Fluor family was mostly published back in 1999. Organelle things are described on the bottom right here. It turns out that some drugs are fluorophores and some fluorophores are drugs. And in fact, the whole chemical industry got started because of the modern chemical industry got started as a consequence of William Perkin trying to purify or synthesize, not clear to me which, I've read some of the history, quinine out of tar, basically, out of coal, or mess with coal. And instead he found a purple dye And he pivoted to that and created and commercialized mauve. And then he and others invented the chemical industry based on that, of basically modern color textiles. And then not long after that, both fluorescence and drugs came to be, and literally the rest is history. A lot of this presentation is going to be various nuances of antibodies and improving antibodies and therapeutic antibodies. I'm not going to give a 3-hour antibody lecture, although this may end up being a 3-hour presentation if I'm not careful. And all I'm going to say here is that you can actually make very tiny, what are called VHH nanobodies now. And that's a lot smaller and therefore diffuses in faster. It's also, if it's 1/10 the size of a standard antibody, and it's simpler format of just a single polypeptide, that's going to cost much less, even less than 1/10 the size of the molecule, to synthesize and store and ship and do everything and use and do everything with it. It does mean that there's less places on it to stick fluorescent dyes, but I'm going to argue that you could stick what I'll call a custom tail onto the antibody that would then have the right number of dyes at the right distance from each other, and maybe the tris NTA nickel attachment also at the right distance for each of them, so that you get an incredibly bright little tiny nanobody for diffusing in and then washing unbound stuff out of your spatial biology experiment. More antibody structure things, my suggestion for custom tail. Nanobodies, one way to make nanobodies is by injecting antigen and an adjuvant, which usually helps boost vaccines, into llamas, or in this case alpacas. I'm going to argue that we have the genomes, or we have the B cells from a bunch of alpacas. You don't really need a giant farm building of 1,000 or whatever number of alpacas, camels, llamas, rats, mice, rabbits, and so on in 2025. You just need to move a bunch of B cell DNA sequences into the lab and use recombinant methods. So I'm not advertising for this company in many ways. I'm advertising that we should eliminate giant stupid farms like this in favor of doing things recombinant antibodies in the lab, and conceivably AI-based stuff, which requires excellent data, which requires recombinant antibodies and sequences in the lab and data of how good these things are. And so we'll see how the whole AI antibody story plays out. Okay. A nice little summary after BioTrac, but before I wrapped up the presentation on nanobodies. I showed the IHC slide, immunohistochemistry slide, in an earlier slide, HRP, peroxidases, alkaline phosphatases. These are all historical methodologies to get this to get the antibody staining working. More about peroxidases, and then it turns out spatial biology intersects with mass spectrometry, in particular with APEX, and now APEX 2, APEX 3, improved synthetic versions of peroxidases that can work inside live cells. HRP, in terms of price, you can buy it at the supermarket as horseradish, as a major component of horseradish, and then can still be purified at your lab bench. Probably simpler to just make get pure stuff by recombinant methods. Although simple recombinant HRP needs some funky carbohydrates in order to work right. That basically the reagents are out there to have synthetic peroxidases, and then you can conjugate that, or even at the recombinant level, stick it onto a protein or an antibody or an antibody. Here's yet another timeline, switching from antibodies and B cells to T cells for a second, that the T cell world has been advancing over many, many decades. It took a while to realize that the thymus was where T cells get made. That's why they're called T cells. A lot of stuff has happened in the last couple of decades. I've been in the CAR T cell world labs twice, and now I'm back to Microscope Core Manager. But the therapeutics all need diagnostics, and so one of the questions are, what are the antigens that are on typically the surface of a target cell of a cancer cell, what I refer to as the oncosurfacome, but not on the expressed at high levels on any normal tissue, because you don't want to destroy the normal tissue if you can help it. Again, back to timelines, recombinant antibodies. Occasionally the slide deck is not perfectly in order because of a little bit lazy part on my part, but you can read slides in whatever order you want. That detector, and I pivot occasionally and then pivot back to antibodies later, that the cameras and other detectors are actually quite good. But different cameras and different detectors are needed for different applications and different price points, and now you can, if you want to buy me a bunch of Quest 2s for my core, I would love to do that, and also the stuff to get them all together, and then the faster, newer computers to deal with the data deluge. That'd be fabulous. As a microscopist, I like to point out the resolution limit of various microscopes. So this particular graphic compares confocal CLSM to a bunch of other methods, and basically CLSM is great, was the standard of care for a long time, and then various other acronyms have come into being that improve resolution in various dimensions to some extent. But typically things like the Palm Storm methods on the right in DNA Paint, that they need thousands of images acquired, or thousands of frames acquired to generate one super resolution image. And go, okay, how long is that going to take? Well, 10 milliseconds times 1,000 images, that's 10 seconds, that's not so bad. But if you need 100,000 images, that's going to take a while, and then you need to deal with the data deluge from that. And then you need to decide, are you going to store that data or not? Not storing the raw data means you can't go back to it. That's bad. You can't actually prove to the reviewers or to the readers of your manuscripts or the reviewers of your next grant. They actually did an experiment, and so there's a bunch of issues with that. In the bottom left, I mentioned some things about the new GPUs, or the not so new GPUs, can help, but you have to code for those, and then all of the best software developers now are doing AI stuff, and so getting things like getting the best version of the spatial localization things, so you can localize spatial biology stuff at the nanometer scale, signal protein molecule scale. The size of a immune synapse I started off with is about the gap between the two cells, about 10 to 50 nanometers. And you need to know, is the protein on the T cell or on the tumor cell, and is there post-translational modification on CD3 zeta and various adapters and the whole signaling complexes. So you'd like to have the resolution, or you'd like to at least be able to have the detectability and ideally quantitation. That's not trivial. Okay, so classic confocal microscopy, the resolution is about 3 times bigger in Z than XY. I give some math over there, blah, blah, blah. I'm a huge fan of spatial deconvolution, which now can be done on GPU if it's coded right and the algorithm is correct and the post-processing and the data out are all done right, that you can dramatically eliminate the background haze of a standard wide field image, which is what most of the spatial biology commercial sources use, and also not only get rid of the haze, but move the photons or the photoelectrons essentially from the detectors back to the right focus point. And then that gets you more better data, and that's good. And then you can iterate or do by erasing off the current round of antibodies and put in new antibodies and detect more things. So I like this example, and deconvolution also helps confocal. And now it's more or less instant gratification with the latest GPUs. This slide image was taken a few months ago on the Leica confocal microscope I managed. The top is somewhat fuzzier compared to the bottom. The deconvolution was pretty quick on my 2060 or NVIDIA 2060 or 2080 card. It would have been, or M6000 card, it would have been near instant gratification on the RTX 4090, a couple of years old, 90 teraflops. The new NVIDIA RTX 5090, I'm recording this on January 19, 2025. The price of the NVIDIA RTX 5090 Founders Edition was announced a week or two ago as $2,000 list price, but availability in terms of checking out the NVIDIA website and ordering one, good luck with that when it's formally launched and commercially available at the end of January 2025. And then lots of OEMs will have the variants of the 5090 card that are bigger and have more fans and are higher price. And at some point, the price, somebody will charge probably $3,000, and that'll be readily available because not that many gamers will want to spend $3,000 on that card. It may take a month or two before the price equilibrates to the right place there. But be able to buy something that turns out going to be rated at about 105 teraflops versus about 90, or 85 or 90 for the 4090. Back to fluorophores again. I could have shuffled the slide deck a little better. There's a whole bunch of technologies out there. I kind of like this Starbright stuff, the academic version of P-dots or polymer dots from DT Chu's lab was published recently in Science for Science Advances. So I look back at that, and Starbright is kind of a cool technology, although about 20 nanometers, a little bit bigger than I'd like. But if it's also brighter, that would be cool. And resistant to photo bleaching. I'm a big fan of Brilliant Violet 421 down here at the bottom left. And then IBEX is one of the many acronyms for this one is iterative bleaching, and then repeating, blah blah blah. We'll have more about IBEX later. Andrea Radke, one of the major organizers, essentially the staff scientist, former postdoc, former grad student at NIH, recently moved to Leica, but is still involved in the IBEX stuff. And then I really have pet peeves about my user base and about the way a lot of researchers do shit. And so I have a little quote at the bottom of old antibodies die, please throw them away. And then I have another quote, probably somewhere in the slide deck, mentioning please do not buy crap from Santa Crap. A lot of researchers, everybody should know what Santa Crap is in terms of bad antibodies, but if you can't, you can always email me. I'm also a fan of what's now often called label-free imaging or bright field imaging, and then certain ways to do fancier illumination and do some quantitation. I could have organized this back with the other HRP tyramide stuff. Thermo Fisher recently launched Allura. It's a little bit pricier and than their previous Super Boost kit, but $6.50 for Super Boost, $10 for Allura. The color scheme or the fluorescent dye available scheme are about the same. If a particular Allura fluorophore is, let's say, 3 times brighter but 1.5 times the cost, 10 divided by 6.5, that would be a win. That could be a win. And then you could mix and match things where the Super Boost kits or the other company tyramide stuff wins compared to this. Of course, you have to spend the time and money to buy all these freaking kits. That's going to be a problem. And then the Allura kit is basically tuned for a particular Thermo Fisher EVOS S1000 spatial biology microscope in a box. And so the performance of Allura on the E1000, on the S1000 versus on a good quality fluorescence microscope remains to be seen. So we'll see what happens with this. And again, more on the tyramide stuff, and I'll emphasize that it's amplification. It may not be linear, and then it works by primarily the activated tyramide radical from HRP plus H2O2 plus fluorescent tyramide creates a tyramide, catalyzes creation of a tyramide radical that diffuses a short distance, let's say 50 nanometers at most. It depends on what's in the solution. And then either makes a covalent bond with a tyrosine residue on the surface of protein near where the original antigen primary antibody HRP stuff was. Often that tyrosine will be on the antibody or on the HRP, or near the antigen, sometimes near the on the tissue or cell. But if there was a tyrosine desert, there wouldn't be any signal. Or if you strip off all of the antibody HRP from round one and do this, that you're going to disappear most of the tyramide dye that you've just covalently deposited in the sample. So if you have a tyrosine desert, that's bad. If you had a completely coated tyrosine surface, optimized amount of tyrosine, that you could get more signal, and that would actually be kind of cool. You could do very beautiful fluorescent microscopy on that surface, and you could do it with TURF, a particular imaging excitation technique, and get incredibly good data with no background. Whether it's going to be cost effective for what you need to do, and whether you can multiplex that, who knows? Okay. Label-free microscopy, again, my slide deck gets sort of desorted, unsorted occasionally. I'm not going to sweat it. This is holotomography, which is available from two companies, NanoLive, and this one is from TomoCube. A nice pipeline. I put the reference, just the DOI at the bottom right, of after you get the data, you got to do shit with it. Multiplex stuff, I'm not going to read the slide, but the middle one, the middle paragraph is the IBEX reference, is a IBEX reference. You can just look for IBEX and RedKey in PubMed to find more stuff. And then I will point out that you got to be careful about reagents. You really do have to look at the data sheets and the warnings, and say in Nature Protocols when it warns you about shit that you need to pay attention. Lithium borohydride, which Andrea uses in IBEX, or sodium borohydride used classically. If you mishandle the bottle, it could explode. That would be bad. So don't do that. Find out from your safety people what the right thing is. Make sure you do that stuff either in a chemical hood, or know from Andrea and from the vendors that the lithium borohydride stuff can be handled safely on, let's say, the cell dive system from Leica. But it's your responsibility not to blow up your lab and possibly hurt people, and break equipment and catch the building on fire and all sorts of things like that. This was a nice little review of a bunch of the different multiplex methods. More multiplex. I'm a fan of smFISH, multiplex error robust DNA or RNA fluorescence in situ hybridization. The VizGen, commercialized by VizGen, spun out of Xiaowei Zhuang's lab to do single molecule RNA fish at scale of let's say hundreds to thousands of RNAs in a relatively cost-effective manner. It's not not cheap. One slide, several $1,000s, but several thousand answers. And then scaling that to clinical, that could be a challenge. But presumably you only need a few targets in the clinic. It is as spatial biology, I'm a huge fan of measurements, of morphometry, of quantitation. So I included this page with a bunch of kind of classic things on my library. The video, I don't know if I have the video, if I may have given away my video microscopy book in the course of various moves, but I do have the 4 on the right, and then the center panel is from the appendix of video microscopy. This is again the same slide deck, and then some more things on the right. More morphometry. It's not like spatial biology has actually discovered any new methodologies for cell-to-cell, nearest neighbor, blah, 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 or within the cell, various parameters, and you can do this on any organelle, typically on the nucleus and the cytosol. But it's all classic stuff. Some of this originated, again, sort of post-World War II, or maybe even during World War, a little bit maybe during World War II, of things like finding tanks hiding in a forest, or now in 2025, finding tanks and surface air missile sites and anti-aircraft guns and drone launch sites in a forest in Ukraine and Russia. Okay, you're going to see a whole bunch of stuff from Brian Kruger. Rather than reading the slides, I'm gonna just flip through those and Brian Kruger and other people from LinkedIn, along with the links and the text. You can check this out by just stopping the video, or by checking out the PowerPoint, the PDF version I post online. That was the end of my BioTrac talk. It's now at 1 hour. I have a lot more slides now, so I'm gonna just roll out and do more things. The original version was, or the somewhat supersized version from original, is currently BE Press 98. This longer presentation is on as a PDF is number 99. The original video that got truncated is 100. That's why I'm re-recording. More web pages, and then a long time ago, I thought about, well, how do I tell different cell types apart in, say, a tissue section? And the pathologists were there way before me. They keratins, and it turns out that there's a whole bunch of other intermediate filaments, proteins, and I'm going to say that those are all good candidates for particular cell types in any given tissue. In normal tissue, all bets are off, actually, when it comes to cancer, or some bets are off in terms of cancer. So any particular cancer often has is a still more or less play the same protein molecules as the normal tissue, but certain advanced cancers have, quote, de-differentiated or even completely pivoted to a new phenotype. Let's say a B-cell lymphoma. You kill off almost all the cells with CD19 CAR T cells, and occasionally the patient will relapse. Instead of ALL, acute lymphocytic leukemia, will relapse with AML, acute myeloid leukemia, or lymphoma. Essentially, a cancerous B cell turned into a cancerous macrophage or myeloid cell. And that often involves change of intermediate filaments, along with the oncosurfacome, and then you got to tell that from the normal surfacome. Back to some Keith Wharton stuff about spatial biology. He argues or posted this comment about 3D histology from a bunch of pathologists. Well, good. I mentioned at the beginning of the deck that not only cancer and autoimmunity and infectious diseases like COVID-19, but GLP-1 agonists. And so Novo Nordisk in the summer of 2024 became the most valuable company in Europe on the basis of it's in a leading position for GLP-1 agonists. And so obesity can be treated, and I'm going to say that spatial biology should be looking really hard at GLP-1 agonists and other players in the obesity / diabetes market. Diabetes, there's two types of obesity, of McDonald's diet, and type 1 diabetes, which is an autoimmunity. And so we should be paying a lot of attention to this. But turns out GLP-1 agonists may also suppress all sorts of different addictions, like drug addictions, smoking, tobacco, and drinking, and gambling, and then may also improve certain types of brain disorders, of mental illnesses. And so if we will need to get cheaper agonists distributed to a lot more people, get it around the world, have some, it doesn't do us that much good if we can cure fatness in New York and LA and Baltimore, for that matter, if we're not also helping Central South America, Africa, various parts of Asia, various minorities in North America, U.S., Mexico, Canada, blah, blah, blah. Okay, so think about, when you think about spatial biology, think about the opportunities for GLP-1 agonists, other agonists, antagonists of the alternative of the various pathways and steps. And so basically all of medicine can likely be improved by spatial biology. Okay. One of the things I did not feature back in the first part of the slide deck was the filters that go into the microscopes that give us clean, no background fluorescence. And so I stuck in this slide that says they've delivered 78 million filters at say $100 a piece, list price, and therefore they've made a whole lot of money. So 78 million, not nearly what McDonald's has sold in burgers over the last many decades. But also, filters do not cause obesity. They inform spatial biology, regular microscopy. I'm a big fan of spectral imaging. There's a lot of stuff that you can measure out of a microscope. I'm not going to talk about them. And then Andrea Ratke and a colleague published, was this Andrea? My apology, look in that. It might not have been Andrea. And you have to look kind of, oh, maybe I did. But the references are at the bottom there. Actually, this was a CAR versus bispecific or BITE paper. My apology to the authors for not explicitly putting them in, but the references are there. Maybe a little bit tedious to find this one. Of the top immunology medicines, this is not immuno-oncology, this is immunology, kind of, or on the left is immunology. And I find it, so anti-IL-4R alpha, I believe, is a asthma and maybe hair, maybe scalp therapy, and then Skyrizi is a IL-12 inhibitor, I believe, and then there's just a whole bunch of stuff in here, right on the left. $23 billion for that top 15, whereas Keytruda at the top right there is already right around $25 billion a year, and is way ahead, something like a factor of 2 or 3 ahead of Opdivo from Bristol Myers Squibb. Again, going back to the beginning, the first slide of the deck, the two companies were tied until Bristol Myers Squibb screwed up a set point of a pathology read that basically was using the simple immunohistochemistry technology that has now gone to multiplex with in spatial biology. So Keytruda, $25 billion, greater than the greater sales than the entire list of the top 15 immunology, non-immuno-oncology molecules there. All of which benefit or would benefit in 2025 from more information by spatial biology in terms of any given patient biopsy sample or mouse model or in vitro sample. Okay, back to GLP-1, a lot of stuff going on, a lot of money, blah, blah, blah. And then I mentioned here that short bowel disease was actually FDA approved a while ago for using GLP-2. And so there's not just GLP-1 and its agonists or GIP. GLP-1 and 2 are out of the same polypeptide, which is pre-pro-glucagon, and a given cell may either chop that up into glucagon or into GLP-1, GLP-2, and potentially other active peptides. I may have a slide on that later. But basically, there's a lot of money here, and spatial biology can play a role. Back to microscopes, that there's a whole sort of circular chain gang here of stuff. Future stuff. It turns out that photoacoustic imaging at the just below center here, that works on the basis of things like hemoglobin and myoglobin and possibly cytochrome C, but also can use exogenous chromophores. And it turns out that things are really good for photoacoustic imaging are really bad for fluorescence, and vice versa. There is some quantum yield of fluorescence versus quantum yield of essentially heat and ultrasound. That you could use the same reagent for both. Probably indocyanine green or ICG might be right in the sweet spot where it's doable now, although not optimal. Probably do better with a really good fluorophore on one part of the thing, of the molecule, and a really good photoacoustic chromophore on another part, or on the identical location on two different molecules. Inject one or both depending on what the application is. Back to label-free microscopy, quantitative phase microscopy, QPM, or quantitative phase imaging, QPI. There's a lot of history here. Again, this is not only a history of spatial biology presentation. And so the graph at the bottom center shows the number of publications over time according to this a particular group. And the idea is that ideally it's quantitative, right? But you don't need to add labels, you don't need to amplify, you just need to have the right hardware or the right software. Going back to Chakra and Lance Taylor's Uttam et al. 2020 paper that was the first spatial biology paper, first one that used the term spatial biology in our modern context. Ron Germain at NIH has published a bunch of different acronyms leading now to IBEX. A little bit more details of more automation. Some pretty pictures from this. Again, my point of number 1.5H, I deliberately repeat stuff in the first half in the first part of the presentation and the new stuff. Tyramides have all sorts of cool things going on. People often reinvent the wheel without citing older literature. And the reference for this stuff is at the top left. I've been into slide scanning a long time. I did not put my favorite tiki goddess mascot picture on the slide. It's okay. Of automation of slide scanners and workflows. Some questions about at the cell level of things happening, which are, again, spatial biology is kind of all of biology minus molecular biology. Yet another quantitative phase imaging, or QP mania, so I kind of nickname it, of at the top left, and then things about the refractive index and the density of cells. This was kind of, the sushi thing was on the left is kind of cool. Of look at this kind of reverse contrast, negative contrast of things, which which for some reason reminded me of a clever clinical or n equals 2 volunteers. Hopefully the PIs injected themselves with a fluorescent solution, and then took videos, and the reference right there, which reminded me of DICE, which was invented by Elizabeth Hillman et al. And is the top center of some mice. And I should mention that for the clinical stuff here, that radiologists do things like perfuse in radioactive isotopes for various purposes, or CT contrast, CT or MI contrast, usually CT contrast agents, PET contrast agents, into people. You can actually do it by fluorescence, at least in that case. More optical clearing. This was a 2024 paper that made international news. I'm like, yeah, but buried in the paper are some things that may have worked better. This is a little classic history about Albert Kuhn's. This is the market as of, according to some marketing company back in 2023. Table I generated for BioTrac in 2023. I forget whether I handed this out or not. So again, this is a list of stuff I put together for BioTrac 2023 of the various players, including slide scanners and a little bit of software stuff, some of the hardware, pre-merger of VizGen and UltraView and other company, and NanoString being acquired from Bruker, blah blah blah. Yet more measurement stuff. This was from a Visio, left is from a Visio farm slide. Who knows what parameters are going to turn out to be statistically significant in a given publication for a given lymphoma, sarcoma, carcinoma, autoimmune disease, with a particular set of markers, and then the whole idea of what are the statistics that you're doing this need to be thought through. A lot of people, I mentioned down here at the bottom left, circular statistics. A lot of people screw up and assume that you should just do a conventional t-test or ANOVA, blah blah blah, using linear means. But if you take the average, and if you take the average of 0 and 180, that is 90 if you're in linear space, regular statistics. But if you're on a circle and that's 0 degrees and 180 degrees, the average of that is not 90 or 270 degrees, but nothing. But if it's 0 and 179 degrees, it's going to be 89 and a half degrees, but that also means almost nothing because the vector is going to be tiny and also kind of weird. So you got to be careful about these measurements. You got to think about context of scalability. Same deal with texture, which I had on an earlier slide. The Golan Lab and collaborators published back around 2012, back when I was doing something kind of spatial biology-ish, called Immunoscore, that is kind of, I think, peaked but maybe gone down in terms of popularity, or maybe the terminology has moved on, or people just don't cite the earlier stuff. Back a decade plus ago, when Immunoscore had come out, Immune Contexture, blah blah blah, that I had found some papers on immunotopographic mapping, and One of the papers was authored by Thomas Grogan, who turns out to be the founder of Ventana, who was the founder of Ventana Medical Systems. He's a pathologist. He also, and then Ventana got acquired by Roche, and I maybe renamed to Roche Medical. And they sell slide scanners and certain reagents and lots of kits for pathology. And here's his book, his autobiography, including a segment about him. Well, it is him. The last part is about him being a cancer patient, and I encourage checking it out. Okay, I mentioned again, my slide deck has a certain amount of repetitiveness. People should normally be using these 1.5H cover glasses on medium and high power objective lenses or high resolution lenses, and probably use it all the time, because you may start out thinking you're going to do things at low resolution, but you may want to move it onto higher resolution microscope. In fact, I'll argue that maybe 40x.95 NA objective lens would be right here, would be a better choice of objective lenses than sort of conventional, kind of either lazy 20x.5 NA, or even lazier 20x not cited in the method section, either model number of the objective lens or numerical aperture of the lens. And so some people use 20x.7 NA objective lens. Some systems have 0.5 NA, some have probably 0.4 NA, but higher resolution, as long as you can control glare, and I had that in the earlier part of the slide deck, going to get you better resolution if your pixel size is small enough, and your cover glass is the right type. Okay. I'm a big fan of sensors. I had a whole slide about sensors earlier, but I wanted to sort of give a back history slide on the invention of CCD sensors. Ultra cool. This is a beautiful technology spun out of Bell Lab, or invented by Bell Labs, and then all sorts of different variants, and of great importance in terms of price. The CMOS technology is much simpler to make, to do in a silicon fabrication center, just like CCDs and RAM chips. CMOS is straightforward to manufacture. CCD is more complicated. Okay, back to fluorescent dyes and fluorophores. The details matter. Basically, if you're going to put fluorescent dyes, or for that matter, antibody drug conjugates, and I mentioned earlier in the slide deck that some drugs are fluorophores and some fluorophores are drugs, and so considerably the same fluorescent antibody could be therapeutic, or the same very expensive therapeutic drug conjugate could be detected by fluorescence, which means you could do fluorescence microscopy directly off the biopsy and see what's going on. More on HRP, and I really like this paper from Llewellyn Smith Lab on full thickness hole mounts, or hundreds of microns thick. But you need to let the horseradish peroxidase antibody conjugate get in. You got to get the substrates in. You got to wash out everything that's not bound. Well, you got to get the stuff out, the unbound antibody out, and then you got to get the much smaller, let's say, diaminobenzidine or tyramide in, along with the H2O2. Not much point in just labeling the outer surface of or the bottom surface, the top surface, if you're trying to image the whole thing, and then it'll turn out light sheet fluorescent microscopy is a beautiful technique one of the beautiful techniques, if you have the machine, to image these thick samples. There they are in the same author, same study, having done DAB immunohistochemistry on it, 300 micron sample. With respect to immunostaining, a lot of researchers don't do enough wash steps, or they do a bunch of wash steps but let the sample dry out because they're they get a phone call, or they get a text message, or they decide to check their email, or they go to the bathroom and they're not paying attention. Sample dries out. Primary antibody is now stuck to the sample forever, never going to wash out. They do a bunch of washes. They may not be consistent day to day, and they get crappy data. So Richard Bury published this book over a decade ago, and he also had presented at a histochemistry conference I attended, and I got his PDF poster, and he reports that 7 rinses after the primary antibody led to better staining than very few rinses. And as I say down here, well, okay, so if 7 is better, is great. Would 9 or 11 or 13, 15, I don't know, continuous flow, or is it the total volume? What's going on here? So could he have done better if he extended this to 9 washes, 11 washes? And then I make the point in a little box that you don't want to let the sample dry out. Bad things happen, especially if you let it dry out while you have your full load of primary antibody on. That would be catastrophic and be completely useless data, useless slide to image. But if you let it air dry for even a second in here, kind of like letting your peanut butter and jelly open-faced sandwich fall on the floor of, if you count to 5, is it okay to eat? I'm like, no. But good luck with that. And then it turns out that there's some more issues with after secondary antibody. And then not on this slide, but I'll say verbally, when you're doing fluorescence, you should always put the DAPI in with the secondary antibody, or even with the primary antibody, not by mounting media that contains DAPI or some other DNA fluorescent counterstain for the nuclei, because that raises the background. So my apology for not finding some space on here to point that out. But we got it verbalized anyway. All right, well, I do put it on the next slide up here, like to use it by the right stuff. 2 components. Prolonged glass is ideal as the mounting media for standard high resolution microscope 1.4 NA, refractive index 1.518. Immersion oil, because that's matched to the glass of both the lens and the cover slab. Again, 1.5H cover glass thickness is normally going to be the right stuff. The prolonged glass is liquid. It needs to solidify. You need to let the volatile compound that keeps it liquid needs to outgas, and then a big cover glass needs more time for the stuff in the middle to get out. And those are all just issues you got to deal with, right? So 22 by 22 millimeter cover glass versus, let's say, 14 by 14 if you could find one. Going to take longer to get out. You can't seal the edge of the slide. The prolonged glass will solidify and take care of that for you. People sometimes just use a protocol precisely as written without thinking through the whole process. And so I had a colleague who insisted on cover glassing their RNAscope experiment because the stupid company protocol said to do that. And I'm like, just stick it in an open face imaging dish and just perfuse it, just wash in the stuff you need, wash out, and then erase the first round and then stick in the second round. And they're like, no. And they didn't do it that way, and they had trouble. The sample moved around. They had a very tiny sample, so it was a pain in the ass to find it. It kept moving, whereas if you do things simple, it would not move. You put the dish on the same way every time. You dial in the same stage coordinates, and then it's there. So you can either do things efficiently or horribly inefficiently, and so don't just take protocols as the gospel according to some stupid company. Okay, I'm a big fan of computers are now fast. Fast is good. Instant gratification is the goal, or should be the goal. 150 teraflop per RTX 5090. Sadly, that was a guess on in the computer gaming online stuff before the official announcement by NVIDIA at CES 2025 in early January. And so the rating at the moment of the NVIDIA Founders Edition card, $2,000, is actually 105 teraflops. They didn't bother updating the slide deck. It may be that some of the OEM vendors, higher price, more fans, more cooling, will be able to juice things up, or if you are willing to take the chance of a couple $1,000 GPU getting killed off by trying to do what's called overclocking, you might be able to increase the speed. But a simpler solution, if the RTX 5090 is small enough and you have a big enough power supply, is just stick 2 in to the same PC, assuming you have enough PCIe Gen 5 slots on it and a big enough power supply. You could just put in a pair of 5090s and go 105 plus 105 teraflops is 210 teraflops. That would actually be pretty awesome. And then it's really a question of, is your code written to take advantage of having 2 GPUs physically, and can the software, let's say GPU spatial deconvolution, take advantage of both of those? And it may be that if you're doing 2-channel Z series, that you could be doing one of them on one GPU, the other on the other GPU. If you're doing 5-channel Z series, you do 2 and then 2 and then 1. You'd like to actually ideally do spectral and mixing simultaneously, or alternating rounds with the deconvolution. So basically more computing power, more RAM on the PC, the right CPU in terms of price performance to control all this stuff, the right RAID array for the NVIDIA the solid state drive array, NVMe drive array, could be the speed essentially of the PCIe X16 Gen 5 slot, which is about 60 gigabytes per second. And that's about half the speed, it turns out, of the current generation DDR6 or 7 RAM that you could get pretty much instant gratification. And then if you're on a network and everything is working spectacularly and you spend enough money and recognize it's going to be obsolete in 4 years, but fast for a decade, maybe more. And so two or 3 rounds of obsolescence, but it doesn't matter, it's fast. That spatial biology should be, we should have hardware that's instant gratification, and they get back up all the raw data. Here's the table version of this stuff. Again, the GPU actually really 105 teraflops probably, so 210. But if you had a big enough computer case, you could put 3 of them in and get to 315 teraflops. That'd be good. RAM is more expensive than these drive arrays, so maybe invest in 512 gigabytes of RAM. That's a lot of RAM, but not 2 terabytes. And then these, let's say the high point PCIe Gen 5 RAID array would be crazy fast, 56 gigabytes per second. I mentioned 60, whatever. Okay. Peter Sorger was the keynote speaker for the BioTrac conference, and he pitched that 3D histology. I had an earlier slide of Keith Wharton Jr. saying the same thing or similar thing, that SAMP biology is 3D, or real biology, live samples, are 4D, or 5D if we include multiple wavelengths. And you can think about 6D as fancy advanced technologies, or F techniques like fluorescence lifetime imaging, microscopy. That you wanna get the most information out of samples in context, and 3D is good. Again, this is nominally a short history, or not so short history. I came across this slide well after the BioTrac conference, giving a multi-page history that I've squeezed into one page here of going back a long time of vaccines and so on to a couple of years ago, right? And things are progressing even within January 2025. Ian Wilkinson is another person I follow on LinkedIn. I see a bunch of slides from him about antibody issues. It's good to, I'm happy that I know this stuff, and essentially this is a background, gives me a background of, I can go find many of Ian's posts from my slide deck here of a whole bunch of either good or bad things happen with regular antibodies, which is why I had antibody stuff for in lab use back in the first part of the slide deck. More from Ian about therapeutic antibodies in mice or more usefully in people. Lots of text. I love recombinant antibodies. A little bit of timeline about some of this stuff. I'm a big fan of recombinant antibodies. We should be using those. They should be better and ultimately cheaper. No more dead alpacas, llamas, goats, donkeys, rabbits, mice, rats. I don't really care much about mice and rats. Rabbits in certain places like Australia have overrun the continent, but are not generally the source of the commercial antibodies. I mentioned earlier, don't buy reagents from Santa Crap. Don't buy any reagents from Santa Crap. Kind of duplicate of that slide, or more from Ian about a very cool receptor, FcRn. Variants on these more Ian posts on antibodies, the kind of flipping back to nanobodies, and also turns out not only llamas and alpacas, but also sharks have sort of mini versions of antibodies. Actually, humans also and mice have a few of VHH, llama, camel, alpaca-like antibodies. Ian posted about mini proteins and then some AI stuff. Hexabodies, more for therapeutic purposes. If you want to have complement do its thing, you want an antibody that, when it gets the right place in the body, can turn into a hexamer. That's just the way that the C1q part of the complement pathway does its thing. More on complement activation, more on engineering antibodies and what's the right variant antibody in terms of carbohydrates and other things in order to treat HIV. He had a provocative LinkedIn post about, does the field really need to humanize antibodies in case of therapeutics? And the answer is, it probably depends. A public project funded by various players, probably NIH and the EMBO and a bunch of other players are spending money to have mostly academics validate antibodies with a particular protocol. So this is some publications about that, and a LinkedIn post, a text from John Gordon on the left. I'd like to see them just focus on modern recombinant ones, possibly. Like, why bother wasting any money on, say, Santa Crap antibodies that are mostly not going to work, or if they work, that's more chance in that lot number, not that the company, you should buy any things from bad companies. More Ian Wilkinson. He's put an Excel file on to help people deal with data of antibody sequences, therapeutic antibody sequences. Kind of fun, a nice tool. I'm not sure Excel is the ideal tool, but it's out there, and people can then turn it into other data sources and do AI blah 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 on that. Ian is CEO of a company to make proteins and antibodies, and has some tips, some marketing plus useful academic tips, some stuff about alternatives to antibodies, and antibodies being special small versions of recombinant versions of antibodies. One of my favorite papers on the topic of nanobodies was this group that's spun out or affiliated with Roche, and actually there's a biotech company now separate from Roche that has these libraries, maybe making new libraries. You can check out the papers, you can check out the forward references in PubMed and other places. Our light chains, our and mice light chains have different, any given antibody has either kappa or lambda light chain along with the heavy chain. And so this post from Andrew Bradbury and his colleague at, or collaborator at another company, have talk about the oddities of human versus mouse, kappa and lambda. This slide points out that why are we still using antibodies, especially secondary antibodies made out of mice, rabbits, goats, donkeys, blah blah blah, of every time people do an experiment in the lab, they're they're essentially helping, they have the blood, literally the blood, of a whole bunch of dead animals in their experiment. And maybe we shouldn't be doing that anymore. Some weird things about what can go wrong with antibodies, besides buying them from Santa Crap. Certain antibodies have bad things happen on them, and that they may or may not be QC'd out correctly. And then some terminology in terms of what kind of bad things can happen to antibodies. So even if you validate on particular human cell lines, on a couple of human cell lines, that doesn't mean that other cells in the tissue in health or pathology might be producing an antigen that binds your favorite antibody, but is not the target that it's made for, or the target that you're thinking about, and so you can screw up. This was kind of a fun thing that kind of, you have to look at the details of how many times they dropped the tube with this stuff of thousands, but you can screw up an antibody by dropping it. I thought that was pretty cool. Made it a LinkedIn post, and then I checked out the paper. I'm like, wow, you can screw these things up. I think a much simpler way to screw up the antibody is a grad student or postdoc, or the PI conceivably, screws up and just leaves the antibody out on the lab bench over the weekend, and in a tub of ice, nominally 4 degrees Celsius. It thaws out, it's at room temperature for the weekend. Maybe they do really stupid things, like if there's a windowsill in there, and if there's a lab bench next to the window in their lab, and they leave it in the windowsill, and it gets zapped with light, that could, over the weekend, bright light, south-facing window, could photo bleach things and cause weird photo-oxidation, and just really dumb things. Nobody would put in their method section of, we used this antibody after we left it out on the lab bench over the weekend, or over a holiday weekend, or over winter break, because we're thinking about eggnog and not over winter break, and on not our experiment, and we just screwed up. But we used it anyway, even though it's not going to work. It didn't work. But we didn't think about the fact that it didn't work, because we didn't review what we did wrong. Antibody discovery stuff. This was a nice little chart about how the immune system, especially on the antibody side, works. interesting thing kind of clinically to get rid of bad stuff on antibodies. Kind of cool. I think I wrote the top sentence there of, is every IgG molecule unique? But the kind of, and then I put a LinkedIn post and some comments about complexity. And so I'll give a tube of stuff that's out of a rabbit or mouse or whatever, or out of hybridoma. Any of those things every single molecule might be unique in terms of what you're pipetting on any given slide. Whereas, let's say, recombinant antibodies should be much tighter in terms of distribution if they're made in, say, E. coli or yeast or some human CHO non-human cell line or Hep293 human cell line. Especially that the cell line has been engineered to maximize production of a single isoform rather than crazy weird shit. This was a cool paper that just came out very recently on, quote, the molecular reach of antibodies depending on the antigen or conceivably on the antibody isotope. So IgG2 has some funky behaviors compared to its reach compared to the other IgGs. Some more stuff from Ian Wilkinson about biosimilars in terms of biologics versus biosimilars for therapy, and you can read about that yourself. Cancer biomarkers. So again, spatial biology. A lot of that is multiplex immunofluorescence microscopy. MPEF is multiphoton excitation fluorescence microscopy. And then the middle section talks about some other tweaky things you can do with that in terms of F techniques or advanced fluorescence techniques. I'm not a huge fan of the MIT stuff. It spews out more metadata that into every single image or the XML file that comes with every image that may or may not be useful. Hopefully it's good and consistent and correct and all of this stuff. A bunch of the major players are, I highlighted at the top right, author list of this stuff. And then, so garbage in, garbage out of all of this schematics, blah, blah, blah. Workflows, hopefully the antibodies were all good. The tissue section is consistent. The amplification methods are done consistently. The room temperature of the staining equipment is the same. The antigen retrieval is the same. You don't run out of a batch of antibody in the middle of your experiment. Lots of things that may or may not be captured in the MIDI stuff. Yet another expansion microscopy slide. Again, I could have sorted the slide deck more carefully. I'm not going to worry about it. You can just read about it yourself and just download the PDF and ignore my text or reorder it yourself. Good luck. One of the things that we should ideally be doing is making the data available and making it reusable. And so this is the acronym for that is called FAIR. Again, nominally a history of spatial biology, and this is now a somewhat famous blogger, Eric Topol, talking about the dawn of spatial medicine. I put in the box on the right the chakra stuff. This is a slide that I ignored when I put together both the slide deck and this talk. Advice that I thought would be kind of fun to include here. You can read about yourself. More plex, more technologies. Expansion microscopy can work on bacteria, which are tiny compared to human cells, and you can zoom them up. The bottom right is 1,000-fold volume, which is 10x by 10x by 10x per dimension x, y, and z. And then that 1 micron length bacterium is now 10 microns long, and the barrel is somewhat bigger, and now you can see things better if you've prepared the sample right. More expansion microscopy stuff. I should maybe go back a slide. So the current thing as of today, mid-January 2025, is you can do 20, what Ed Boyne is calling 20 EXM? 20-fold expansion microscopy. Did I misspell that someplace? Single expansion, single round. Yeah, sorry about that. I think it's 20 EXM. It's really, as they talk in the paper, that it's really 15x to 20x, or any value in between there, depending on exactly how you do your sample prep. So you want to be as consistent as possible with that. And then I feature on the right middle text of you're going to control the oxygen environment and remove all the oxygen, essentially, using, in their case, a glove box to because the oxygen can suppress the gelling reagent. Okay. I wanted to put in this slide. This is a table / graphic I have not updated in a while, showing many fluorescent protein biosensors. I think a lot about live cell imaging. How many fluorescent protein biosensors can we have in one cell? How many different cells with different sets of biosensors can we have in a given experiment, say an imaging dish or perfusion thing, or in a mouse or a human or a zebrafish or whatever. And so there's a lot of things out there. A lot of them are kind of crappy. But some of them are quite good, or maybe one amino acid change, several months of one grad student or one grad student well-mentored work could take a mediocre biosensor and turn it into a great biosensor. Maybe just swap out crappy 1996 EGFP for the state-of-the-art M-state gold, monomeric state gold, or a monomeric version of AAU SFP1. In terms of green, M scarlet 3, or the latest variant of that's also even more stable than M scarlet 3. Red fluorescent protein. A lot of things can be measured, quantified in real time. I've been talking about GLP-1. Its receptor is a GPCR, G protein coupled receptor. People have made neuropeptide receptors of fluorescent protein biosensors, including to GLP-1R. The tools are out there. The papers are out there. People can either get the plasmids from Angene, create the sequence for whatever organism, let's say zebrafish, with cell type specific promoter that they want, or collaborate with the authors as well. A recent technology feature in Nature, RNA stuff, precision medicine, or conceivably spatial medicine. Eric Topol's slide a few slides ago, and I wrote out how to think in terms of different scales, in terms of both pixels, voxels, microscope stuff, up to the other large volume world population of 7 billion. I mentioned Santa Crap. Give an example at the bottom right of a paper published a month ago of might be flawed. I would say that that all 1,400 papers of this using this anti-Bax thing are seriously flawed, are completely screwed up. And then it turns out the journal that was published in has 2,800 papers published using antibodies from Santa Crap. And very likely at least one of those, if each use, say, 3 antibodies from Santa Crap, and at least one of them was wrong in every paper, then all 2,800 manuscripts may be bad. Are they going to get retracted for cause? Probably not. And so basically, yeah, the paper that reports fairly well that this anti-vax is flawed, is in a journal that itself has a major flaw. A little bit of pathology-related things here on this slide, and then some 3D stuff. And I like to use the $sign instead of S when there's money involved, and so there's a nice little summary of optical clearing, 3D light sheet fluorescent microscopy if you have the right light sheet microscope. That you can see where the HEC3 GLP1R is in, let's say, an entire mouse brain. But you can scale that up. You could do it on rats. You could do it on marmosets. You could do it on monkeys. You could do it on chunks of human brain, obviously ex vivo, or in some cases biopsies of parts taken out to treat epilepsy or cancer or whatever. And so you want to know where the signal is and so there, the bottom center here, the blue, is detecting CFOS, a immediate early response gene, or IEG, that lights up after infusion of the GLP-1 agonist in the mouse brain in this case. And so that's probably the site of a major site of action. Although it could be that there's little parts of the brain that have important neurons that then receive that signal and send signals off to other parts of the brain with other neurotransmitters and neuropeptides. Okay, here is a recent late November 2024 Nature paper on another neuropeptide, and there's a whole bunch of neuropeptides. I'm not an expert in them, but there's probably 100. There could be more. There's 400 G protein coupler receptors. A whole bunch of them are orphans in terms of not knowing the endogenous ligands. It's likely a fair number of those of that 400, or the X number of orphans that are left, are binding and receiving signals or signal transduction through using neuropeptides as the ligand. And then they go off and produce signals and inhibit or excitatory neurons in the brain, or from the gut vagus nerve up to the brain, whatever. Okay, so there's a lot more things that are going to need to be learned. The approach from that slide for GLP-1 could be used on any of the neuropeptides. And if you don't know what the receptor is, then at least that would tell you where the ligand causes, let's say, the CFOS gene to light up to get transcribed to a high level for a certain amount of time. And then you could considerably do single cell RNA sequencing or look up the current atlases of what GPCRs or other receptors are made in that body part, that brain part. And then deorphanize the GPCR and then make, let's say, small molecule agonists that would, or antagonists that will affect that neuropeptide. And then X number of those could improve GLP-1 and its agonists even more, and cure various things. Okay, back to fluorescent dye, or fluorescent reagent. Fluorescent dyes I think of as the small molecules, like fluorescein or alexafluor-488. I think of the other technologies as fluorophores, or the whole group of things as fluorophores. And then some marketing company claims that the field of fluorescent dye market will be about $3 billion in 2028, 3 years from now. Good luck with that. I don't know where they come up with that number. I didn't buy their report. The growth rate is pretty good. Use research and diagnostics, and then the larger, if you double the scale of your spatial biology experiment and go from 50-plex to 100-plex, that's going to be twofold more purchase of either the same fluorophores or less likely different fluorophores, and probably twice as long to acquire the data. And then I started off the slide deck with Bristol Myers Squibb screwing up a immunohistochemistry set point for their Opdivo anti-PD-1. In November 2024, Amgen similarly managed to botch something about their obesity drug, so some either play on GLP-1 or some other thing, and do certain measurements, and maybe they should have done maybe some spatial biology in mouse models, or more mouse models. It wasn't a set point in this case. It was actually a very funny computational or data thing of someone spotted that there was a hidden worksheet tab in an Excel file that had been made public, and that dropped Amgen's stock price. I thought that was kind of funny and bizarre, and a little bit sad for stockholders, sad for some employees at Amgen, maybe, and hopefully the stock analyst got a raise out of that. And then somebody pointed out that the data kind of sucked, and therefore, why was it part of this whole thing? And then the article basically, or the post on LinkedIn, says, comments that transparency and clarity are crucial. That was very kind of funny. So anyway, this is the end of the slide deck. Thanks for listening, and it may have been easier to just walk through the slide deck manually as the PDF. And thanks for listening. Bye.